Thank you, David, and also thanks to the LFSA committee for giving me this opportunity to present. It's also important to acknowledge members of my laboratory who've done all the work that I'll present, especially Pingyang and Jerry, whose work I'll present today on metformin and Li-Fraumeni syndrome, in keeping with the theme of this session. I'll try to move as quickly as I can because I think I'm the 10th speaker for this morning session, and I'm sure you guys are all ready for lunch, so I'll try to go as quickly as I can. So what I'd like to do today is share with you again our work in the laboratory trying to develop strategies for cancer prevention and Li-Fraumeni syndrome. And this is work that actually was published earlier last year, but I would like to present this also, just to also again to remind you of what we've done and see if we can initiate further thought and consideration for the use of metformin in Li-Fraumeni syndrome. So what we showed is that both the genetic and pharmacologic disruption of mitochondrial function in a mouse model of Li-Fraumeni syndrome can delay cancer formation. And we went on also to do a parallel translational study in individuals with Li-Fraumeni syndrome to show that by using metformin, that we could indeed have evidence of inhibiting mitochondrial function. And also replicate some of the anti-proliferation signaling that we observe in the mouse model associated with delayed cancer. So our interest in this field started probably over 10 years ago when we observed that P53, wild-type P53, can promote mitochondrial function. Just as a word about mitochondria for the lay folks in the audience, the mitochondria can be thought of as the powerhouse of the cell. There's small intracellular cellular organelles within a cell, and can be thought of as sort of the furnace of the cell that burns food that we eat, glucose, using oxygen. And so that's a process is called respiration and results in this process called oxidative phosphorylation to make ATP that is used by the cell for its many different processes. So what we showed was that, and others have shown also with other pathways, have shown that both in the nucleus and within the mitochondria, P53 can promote mitochondrial function, and at least in the nucleus, it can do this through multiple different genes shown here. We in particular studied SCO2 and TFAM, mitochondrial transcription factor A, which are really critical for mitochondrial biogenesis. But P53 also seems to get directly into the mitochondria through very specific molecular mechanisms and interact with the mitochondrial genome or mitochondrial DNA to facilitate this transcription and maintenance for mitochondrial function. And these mechanisms, at least in the models that we've used, seem to facilitate very important physiologic phenotypes such as exercise capacity and could potentially also impact tumorigenesis. And we've shown this that both wild type and LFS mutant P53 can do this as well. So while this again was done in cells and mouse models, we wanted to see if this is also relevant in human. And so a number of years ago, we did a small pilot study enrolling LFS patients and looked at the in vivo mitochondrial function, and we used a technique known as phosphorus-31 MR spectroscopy that involves leg exercise, and we're measuring phosphocreatine levels. And so basically, I won't go into the technique other than to say that we had actually predicted that individuals with Li-Fraumeni syndrome, because they had mutation in P53, that they would have dysfunctional mitochondria. But to our surprise, we actually saw that actually had a faster phosphocreatine recovery time constant, again, which is just essentially a marker of increased oxidative phosphorylation capacity compared to carrier individuals who did not have the mutation. So at this point, we thought that perhaps by if we can modulate or downregulate mitochondrial function in LFS, could that be a strategy for potentially delaying or preventing cancer formation? And so to address this, we turned to LFS mouse model, the famous 172 mutant mouse that Gigi had created a number of years ago. And we took advantage of another mouse known as the polymerase gamma mutant mice. The polymerase gamma is an enzyme very important for the mitochondrial genome. It repairs and replicates mitochondrial genome. And this mouse is available. So we crossed these mice that have slightly dysfunctional mitochondria into the LFS genetic background. And we looked at oxygen consumption or respiration in the cells in which the thymus is a typical site of lymphoma development in the LFS mouse model. And so we're able to show that when we crossed POLG mutant mice in a mutant allele dose-dependent manner, so this is heterozygous and homozygous mutant POLG in the LFS genetic background, that we could indeed decrease mitochondrial respiration, along with just showing here a Western blot, a downregulation of complex 1, which has been shown to be associated with POLG mutation. This is one of the complexes within the mitochondria that are involved in reducing or using oxygen. And we also observed in the plasma of the mice, the blood of the mice, increased lactate levels, again in a mutant, POLG mutant dose-dependent manner, reflecting perhaps a compensatory increase in glycolysis. So glycolysis would be the anaerobic metabolism, not using oxygen. And we saw increase in some of the glycolytic enzymes, glucose transporter, as well as hexokinase 2, again, is involved in glycolysis. So the cells or the mouse is trying to compensate for the decrease in mitochondrial function to try to increase energy production. So we wondered whether there may be other, so at this point, I guess, the important question is by downregulating mitochondrial function in thymus tissue or in the whole mouse, can that affect cancer outcome, of course? And so we are able to show that, again, in a polymerase gamma mutant allele dose-dependent manner, that there was a 40% delay in cancer or increase in cancer-free survival in the LFS mouse. This is the homozygous mutant mice, and 80% increase in the homozygous mutant POLG mice. Again, the LFS genetic background, and as some of you may already know, the POLG homozygous mutant mice actually have a very severe phenotype. It's actually been used as a model of premature aging and have very severe defects, but does improve survival. But the POLG heterozygous mice actually have a very mild phenotype and, in fact, have normal lifespan. So we mainly focused on using the heterozygous state of POLG, so just as a very partial or mild disruption of mitochondrial function. We also, because we had been using mostly homozygous LFS mice, which, again, for the purposes of cancer-free survival time, they developed cancer faster, so we had used those mostly. But also just to show you, in the heterozygous LFS mice, crossing these mice, again, with the POLG heterozygous mice, can still also improve cancer-free survival, although the survival is much longer, up to over a year. It improved this, again, by about 40%, which is what we also observed in the homozygous LFS mutant state. So as additional additionally linking this kind of disruption of mitochondrial function with preventing cancer, we're able to do this correlation between the plasma lactate levels, again, reflecting the degree of mitochondrial inhibition in each individual mice with survival time, both in the POLG homozygous mutant mice as well as the heterozygous mutant mice. There was a correlation between the degree of mitochondrial inhibition, again, reflected by lactate level, and survival time. So besides lactate, we wondered whether there might be other cellular signaling changes that could potentially be used as biomarker of the inhibition of mitochondrial function. Again, that correlates with cancer-free survival time. And so it's known that the mutant mutant P53 can downregulate autophagy. And this is a Western blot looking at LC2, which is the autophagy marker. And autophagy is a process cells use to degrade or get rid of proteins or organelles that are not dysfunctional or not needed. And we would predict that in the Pol-G mutant state, that with disruption of mitochondrial function, that the cells would try to eliminate the defective mitochondria, and so you'd expect to see increase in mitophagy. And indeed, you can see here that when we looked at the thymus tissue, again looking at the tissue where the tumors develop, that in the LFS mutant state, there's decreased autophagy or mitophagy, and that when you cross the LFS mice with the Pol-G heterozygous mutant state compared to Pol-G wild type in background, you can see that there was an increase in autophagy, as you might expect if the cells are trying to clear mutated, defective mitochondria. And we're also interested to see that, that there was a decrease in the activation of GSK3 as measured by phosphorylated GSK3. And this was interesting to us because GSK3 activation is known to downregulate. One of the things it does is to downregulate cyclin D1. So as you can see here with this, when we cross these mice again with the Pol-G mutant state, that there was a reversal of this downregulation of GSK activation, and associated with that, with the increased GSK, you can see that there's also a decrease in cyclin D1 that has been shown to be important for lymphomagenesis. So I'm sure there are many different factors that contribute, but this could this sort of cellular signaling processes could potentially contribute to a tumor suppressive phenotype that we had observed in the survival curve that I have just shown you. So we wanted to do so we wanted to see whether we could reproduce. Now, genetic experiments are very clean, but we would like to use drugs that potentially can inhibit mitochondrial function, so that this can potentially be used in human for treatment or chemoprevention. And so we wanted to see whether we could pharmacologically disrupt respiration in LFS mouse. And so we turned to we wanted a drug that is easily available, and there are plenty of drugs that inhibit mitochondrial function, but they're very toxic and actually are poisons. But we wanted to use something that could potentially be used immediately off the shelf, and a natural choice for that was metformin. And I think many of you, probably most of you, have heard about metformin. It's a medicine that's very commonly used for diabetes. In fact, it's probably the most widely used medicine in the world. It has many different beneficial metabolic effects, but one property it has is to decrease mitochondrial function, and it's thought to do this specifically through inhibiting respiratory complex 1 that I showed you earlier, that actually was decreased in the Pol-G mutant mice. It's a safe and well-tolerated medication, and the great thing about this is it's non-hypoglycemic. It doesn't drop your blood sugar, which is a dangerous side effect of many of the diabetic medicines. Just to mention that there have been a number of observational studies that have associated metformin use in diabetic populations with decreased cancer incidence, though some of these findings there's some discussion around this, whether it's really valid or not. So we fed the LFS mice with metformin in drinking water, and we used a couple of different concentrations. The highest concentration we used was 1.25 milligrams per mil of drinking water. That dosage of metformin has shown to be to result in a therapeutic dose, what you might find in human at the human dose. So it's not a supraphysiologic level of metformin. So the therapeutic dose of metformin in human subjects as well, and drinking, having the mice drink this metformin, we're able to show that there is indeed some degree of decrease in respiration in the mitochondria in the thymus cells, as well as activation of the GSK phosphorylation and downregulation of cyclin D1, again, just serving as a biomarker of its activity in thymus tissue. And the important question, again, is does this affect cancer outcome? And indeed it does. We were very pleased to see that at the therapeutic dose of metformin at 1.25 milligrams, there was a 22% increase in median cancer-free survival time. And that's not as significant as the 40% that I showed you with the POLG heterozygous mutant mice, but it is statistically significant. And I should also mention that the metformin was fed to these mice at beginning after the mice were weaned from their mothers, which is usually about age 4 weeks or later. And so just to remind you that at least in the genetic models where you have the POLG heterozygous mutant mice crossed with LFS mice, that's beginning with a single cell stage that you have this kind of change in mitochondrial function. While here, this would be at almost young, sort of youth age level for mice. I'm not sure what 4 or 5 week corresponds to for mice, but they're reasonably grown up. So having seen this, we're encouraged by these results, and we wanted to see can we also inhibit mitochondrial function in LFS patients if we have them take metformin at therapeutic doses that are used for diabetic patients. And so we had the opportunity to collaborate with Tito Foho and Fazana Walker, who were at that time at the NCI intramural program. And they were interested in examining the safety and tolerability of metformin in LFS patients and wanted to measure biomarkers of metformin treatment. And we were mainly interested in the mitochondrial function. And so we did this part of our study before, during metformin treatment, and after treatment. And just as a schematic, we was a 20-week study. The subjects came to the NIH, and we tested the mitochondria at week 0, which is before treatment, and then they had dose escalation at 500 milligrams every 2 weeks until they reached the maximum dosage of 2,000 milligrams of metformin. That's the maximum recommended dose for diabetic patients. And we then measured the mitochondria week 8 and week 14. And then had a washout period to show specificity of whatever findings there may be. And again, we're very pleased to see that with taking metformin at week 8 and 14, that would be the maximum dose time points, that we could indeed show decrease in mitochondrial respiration in blood mononuclear cells. So this is just primary cells, just isolated from the blood, and then we take those cells and measure oxygen consumption. And with the washout period without the drug, you can see recovery of mitochondrial respiration in the mononuclear cells by this respirometry study. And in addition, in the blood mononuclear cells, we again looked at the cellular biomarkers. And this is again looking at the autophagy, LC32. We're able to show that at least in 2 representative patients at the maximum doses of metformin, that there's an increase in the autophagy marker, as well as activation of GSK3 marker and downregulation of cyclin D1 at the 2 time points that are in red here, compared to baseline and with washout. And the important question now is in vivo, these are all ex vivo cells verified. In vivo, is there evidence of mitochondrial inhibition in the patients? And so then we again use the phosphorus 31 MR spectroscopy, the leg exercise. Again, this is a non-invasive marker of OXPHOS, active OXPHOS in the skeletal muscle of our LFS subjects. And we're able to show that, again, at the 8-week and 14, which would be the maximum treated dose, that there was an increase in phosphocreatine recovery time constant. And the higher number actually is the opposite. It means that the mitochondria takes longer to regenerate ATP, meaning that it has downregulation of mitochondrial function. So we're able to show that with metformin treatment, that there's a delay or decrease in mitochondrial function, and with the washout, that there's recovery with time. And so this, I believe, really represents the best data that we have, that in vivo that we can modulate mitochondrial function in a tissue actually that can give rise to one of the skeletal muscle tissue that can give rise to one of the classic Li-Fraumeni syndrome tumors, rhabdomyosarcoma tissue. And so just to summarize, I hope I've been able to show you that both by genetic and pharmacologic inhibition of mitochondrial function in a mouse model of Li-Fraumeni syndrome, that we can initiate this sort of anti-proliferation signaling that might contribute to this delay in tumor genesis or increased cancer-free survival that we observe. And that it's also, as proof of concept, that it's also feasible to inhibit mitochondrial function and initiate the same signaling in the blood cells of patients with Li-Fraumeni syndrome. And that, and by presenting this data, I hope that to the, I think this is the best community to show this data. I hope that it will promote some discussion and further consideration for a randomized placebo-controlled metformin study, which is really what is needed to show that it is really truly effective for getting some benefit of delaying cancer formation in LFS patients. And in the laboratory, we are continuing experiments. We continue to study some LFS patients that their metabolism and also knocking this mutation into mouse models and trying to further understand about how P53 regulates metabolism. And I just wanted to also mention that although in general what we have reported is that LFS mutations seem to promote oxidative metabolism, as nature will have it, it always has to give, it's always the exception that might prove the rule, but there's always an exception. We actually this past year had an LFS patient contact us who had a lifelong history of fatigue, it's very severe fatigue. And it turns out that when we studied her, she has a very delayed mitochondrial function. So the phosphocreatine recovery time constant was 80. And most patients we've studied is in the range of 30, 31. So, so we are actually studying and trying to understand more about what's going on there. But I just wanted to take this opportunity to mention that if there are any LFS patients, or if you know of any LFS patients who has fatigue, but of unexplained etiology, that would also be interested in studying them to further understand the biology. So with that, I will stop and acknowledge everyone who has made this possible, especially the LFS study participants, some of whom are actually at this meeting. And I really thank them for making this type of study possible and giving it some clinical relevance. And also all the collaborators, Louis Strong, who was one of my earliest collaborators that made this the very first study possible, as well as Tito Foho and Fazana, and of course my laboratory members. Thank you very much.
I think we'll take one question. In the afternoon, we have another therapeutic session and some other things, so it'll be interesting to see how this evolves. So, Arnie, quickly.
Yeah, that was very nice. I had a couple of questions. The first is, what happens to reactive oxygen levels when you reduce redox potential like that with metformin?
So, in general, what we've seen in cells, we can see that if we inhibit respiration, you increase oxidative stress. I think the extreme example would be if you knock out the mitochondria completely, so the cells can't respire. And that actually, we actually did some work with that, and the cells are incredibly stressed. P53 shoots up. So, because I mean, mitochondria is probably the best antioxidant, and I can say this to everyone, anyone who listens to me, mitochondria is the best antioxidant mechanism that you have to deal with oxidative stress in a cell.
And I had one other question. It's my understanding that there are clinical trials going on now for the incidence of cancer in non-Li-Fraumeni populations, in the general population, with metformin. And do you know if those have reported out, or do we know anything that's happened?
So there's been a lot of interest in metformin for cancer treatment, and in general, that has been pretty negative. So far, what I've heard, I really haven't kept up with it, but there is a study that's looking at metformin for age. I think it's just getting started for preventing aging.
There was a study with type 2 diabetics who usually take metformin, and it was anecdotal because some of them take insulin, some of them
Right,
don't,
right,
and
right,
that's a confounding factor.
Right.
But they were trying to sort that out in some way, and I just haven't kept up with it.
Yeah, but I don't think there's any clear data showing that metformin prevents, in randomized placebo-controlled way.